Hi, I'm Grace Shenhar, a urology resident from Rabin Medical Center in Israel and a visiting scholar in Stanford University. Thank you for the opportunity to present my work on rat irritant cystitis. The study was funded by a Stanford University faculty startup fund. Interstitial cystitis, bladder pain syndrome, ICBPS, is a debilitating disorder affecting millions of women. There is a considerable overlap between ICBPS and symptoms of overactive bladder. Patients commonly report symptom exacerbation triggered by exposure to specific dietary compounds. Mast cell dysregulation was suggested as a mechanism. Our aims were to evaluate an acute intravesical exposure in a pilot animal model for 6 environmental exposures associated with bladder overactivity and pain, and a novel agent implicated in mast cell destabilization, and to characterize the dose-response threshold for the 7 suspected irritants. After institutional animal ethical board approval, we conducted the experiment on 7 female adult wild-type rats. We used urethane for anesthesia and then introduced a catheter through the bladder dome to allow pressure measurements during bladder filling and voiding without obstructing the urethra. For cystometry, we began filling the bladder with normal saline for 2 cycles or more until we reached a stable, reproducible baseline pattern. We measured voided volume for each void, practically wiping every void with an absorbent paper and weighing it before and after the void. And as you may remember, absorbent paper was not easy to find in the beginning of the pandemic. We manually removed and measured the post-void residual volumes, PVRs, after each fill cycle. After 2 to 6 baseline fills with normal saline until the bladder reached a pattern, we changed the infusion to one of 7 irritants, 1 per each rat, at dose-escalating concentrations. These were caffeine, ethanol, capsaicin, which is the active compound in chili peppers, citric acid, acetic acid or vinegar, saccharin, an artificial sweetener, and compound 4880, a mast cell secretagogue. Results are shown over time, and dose of intravesical irritant are 0 for saline. Each data point is a complete cystometry fill cycle. Following citric acid exposure, at the very low doses, voided volumes increased, resulting in improved voiding efficiency. Starting at 0.1 mg per milliliter, we saw a reduction in voided volumes, and from a dose of 10 mg per milliliter, higher PVRs, resulting in a combined effect of reduced voiding efficiency. After acetic acid exposure, PVRs were reduced from 0.01% and voided volumes increased at 1%. Then gradually decreased with repeated cycles at the same dose. Intravesical caffeine showed a dose-response threshold beginning at very low concentrations. PVRs decreased, resulting in higher voiding efficiency and loss of functional bladder capacity. Following exposure to capsaicin, there was an initial decrease in PVR beginning at the lowest concentrations, then marked increases starting at 1 micromolar and worsening with time, with subsequent decreases in voiding efficiency. Following intravesical exposure to the artificial sweetener saccharin, we saw a dose and time-dependent decreases in PVR from a threshold of 0.01 mg per milliliter, with reduced functional capacity and increased voiding efficiency. Ethanol is implicated as a cause of LETs via polydipsia and polyuria. When given intravesically, it was associated with a slow, gradual decrease in voided volumes beginning at a dose of 0.1% with relatively stable PVRs, resulting in a loss of voiding efficiency. Following exposure to intravesical mast cell secretagogue compound 4880, we found no obvious physiologic effects on cystometry up to 100 microgram per milliliter. This was a pilot study to explore the effects of intravesical administration of compounds suspected to exacerbate bladder symptoms in ICBPS. Acute exposure of an otherwise healthy rat bladder with no pre-existing inflammation demonstrated cystometric effects for all intravesical agents except compound 4880. This model has limitations. Agents are not necessarily excreted in urine unchanged or in the high concentrations that we've tested. Since the bladder has a thick glycoprotein layer, actual concentrations of urine metabolites near the urothelium are unknown. Cystometric effects at superphysiologic concentrations may actually correlate with symptoms experienced in physiologic concentrations by patients with bladder hypolagnesia, hypersensitivity, or loss of bladder barrier function. Intravesical irritants can help improve animal models of ICBPS. Thanks for listening, and I hope to meet you in person next year.